हेलो दोस्तों, माय नेम इज नेहा। आज हम देखेंगे व्हाट इज द स्ट्रक्चर ऑफ द बैक्टीरियल सेल। लास्ट वीडियो में हमने देखा है मॉर्फोलॉजी ऑफ बैक्टीरिया। उसमें हमने पॉइंट्स कवर किए, साइज ऑफ द बैक्टीरिया, वेरियस शेप्स ऑफ द बैक्टीरिया एंड व्हाट आर द अरेंजमेंट्स ऑफ द बैक्टीरिया. नाउ इन दिस video we will see the structure of the bacterial cell. So I have divided this video in the two parts. So in the part one we will see about the flagella, bacterial parts like flagella, pili, capsule and cell wall. And in the part 2, we will see the cytoplasmic membrane, cytoplasm, ribosome, mesosome, intracytoplasmic inclusion, nucleus, and spore. So we will start first. So what is the structure of the bacteria? So here you can see the diagram. You can see this hair-like structure is called as the pili. Then the outermost layer is the capsule, and this dotted line is the slime layer. Then inside this capsule is the cell wall. Then cytoplasmic membrane, and inside this cytoplasmic membrane there are cytoplasm, mesosome, this enfolded part, then nucleoids, cytoplasmic granules, and ribosomes. And this long hair-like structure is called as the flagellum. So these there are two components, important components, like first is the cell wall and second is the cytoplasmic or the plasma membrane. So inside this plasma membrane there is a protoplasm. So in that protoplasm contain the cytoplasm and cytoplasmic inclusion. So what are the cytoplasmic inclusion in that? Ribosome, mesosome, granules, vacuoles, and nuclear body. So these cells may be enclosed in a viscid layer, which may be a loose slime layer, that is called as the capsule. You can see this bacterial structure looks like a capsule. So in that, many bacteria have filamentous appendages. So these are the filamentous appendages is called as the pili or fimbriae. This pili is also called as the organ of adhesion, and this flagella is called as the organ of locomotion. So now we will start first flagella. So what are flagella? Flagella are the long, slender, thin hair-like structure, cytoplasmic appendages, which are mostly responsible for the motility of the bacteria. So they are 0.01 to 0.02 micrometer in diameter and 3 to 20 micrometer in the length, and they are mainly found in both gram-positive as well as gram-negative bacteria. So this flagella is also called as the organ of locomotion. So these flagella can be seen by ordinary light microscope by using the special staining technique. And in the flagella, they are made up of the protein like called as flagellin, and they are similar to the keratin or myosin. And then this protein mainly responsible for the bacterial motility. Okay, so you can see that this is the structure of the flagella. In that there are three parts: filament, hook, and basal body. Okay, so this filament is the thin, cylindrical, long outermost region with a constant diameter, and this contains the protein called flagellin, having molecular weight ranging from 20,000 to 40,000 Dalton. So, and this basal body contains the rings. So these are the rings like LPSM. So in the gram-negative bacteria contains these four rings. So L means lipopolysaccharide layer of outer membrane. Then P ring is in peptidoglycan layer. S means cytoplasmic membrane, that's semi-position of the membrane. And M ring means cytoplasmic membrane. And in the gram-positive bacteria, only S and M rings present in the basal body. So these flagella may be seen on the bacterial body in the following manner. So what are that? So first is the monotrichous. Monotrichous means you can see in this diagram. So one bacterial cell contains only one flagella is called as the monotrichous. Then second is the lophotrichous. Lophotrichous means you can see, so there are this is the bacterial structure, and you can see two or more than two bacteria, more than two flagella is called as the lophotrichous. And third is the amphitrichous. Amphi means both. So this is the structure of bacteria. You can see both sides there are the flagella is called as the amphitrichous. And last is the peritrichous. Peritrichous means this is the bacterial structure, and you can see many flagella are present on the all side of the bacteria is called as the peritrichous. Okay. This is all about the flagella. Next, second part, we will see the pili or fimbriae. These pili are the hair-like microfibrils having 0.2 to 2 micrometer in length and 5 to 7 nanometer in the diameter. They are the thinner, shorter, and more numerous than the flagella, because flagella are the thicker than the pili. So these pili are mostly present only in the gram-negative cells, and these pili contain the protein known as the pilin, and its molecular weight is range from 18,000 Dalton. And these villi we can see under the electron microscope, and in one bacteria there are around 100 to 200 fimbriae or pili are present. Okay, so these pili join the bacterial cell in the preparation for the transfer of DNA from one bacterial cell to the another cell, and mostly these pili are involved in the conjugation process. Therefore, the pili are called as the sex pili or fertility pili or F pili. So these are the names of the pili. So next we will see the what is the difference between the flagella and pili. You can see the size. When we compare the size in flagella, the size is larger as compared to pili. In pili, the size is smaller. Thickness, flagella thickness is the more as compared to the pili. Then attached to the cell wall. Which part is attached to the cell wall? So you can see this pili is attached to the cell wall. Next, origin. So these flagella originated from the cytoplasmic membrane and pili is originated from the cell wall. Then who is called organ of locomotion? So flagella is called as the organ of locomotion and pili is not. Pili is called as the organ of adhesion. And who required for the conjugation process? Mainly pili help to transfer one material or one DNA to the another bacterial DNA. So mostly pili are involved in the conjugation process. So next, we will see the function of the villi. So mostly this villi plays a major role in the attachment to the surfaces. Therefore, it is called as the organ of adhesion. And these mostly helpful for the transfer of genetic material like DNA from donor cell to the recipient cell. And these villi or fimbriae are the antigenic. That's why we use for the serological test and the preparation of the antisera are the devoid of the fimbriae. So next part we will see the capsule. Third part. First we have seen the flagella, second we have seen the pili, and now we will see the capsule. So this capsule is the main in many prokaryotic microorganisms. So they synthesizes the amorphous organic exopolymers. So what is exopolymers? The polymers that are deposited outside the microbial cell is called as the exopolymer. So this exopolymer is called as the capsule or slime layer or glycocalyx or sugar coat. So these are the four name of the capsule. So term mainly this capsule refers to the tightly attached to the cell wall. Mostly ये जो capsule होता है वो सेल वॉल के साथ अटैच होता है एंड जो स्लाइम लेयर है जो कि आप देख सकते हैं ये जो डॉटेड लाइन है दिस इज कॉल्ड एज द स्लाइम लेयर इज अ लूज structure that often diffuse into the growth medium. So this is the structure of the capsule. You can see here there are two types, microcapsule and macrocapsule. So what is microcapsule? So the capsule layer may be thin of size like less than 0.2 micrometer. This called as the microcapsule. You can see in this diagram, this is the microcapsule having size less than 0.2 micrometer and the thick layer of the size more than 0.2 to 10 micrometer are called as the macrocapsule. Here you can see the structure of the macrocapsule. So next important point is that this development of the capsule requires the favorable environmental conditions like sugar concentration, then blood serum or for the growth in the living host. So next we will see the function of the capsule. What is the function? First function is the protect the bacteria because this is the outermost layer of the bacterial cell. So first they protect the bacteria from the antibacterial agents. Then they inhibit the phagocytosis. Phagocytosis means जो macrophages होते हैं वो process करते host में like phagocytosis process. So they mostly inhibit the phagocytosis process. Then they may provide the protection against the temporary drying as well as they may block the attachment to the bacteriophages as well as they promote the stability of the bacterial suspension and they promote the attachment of bacteria to the surfaces. So these are the function of the capsule. Now next fourth part we will see the cell wall. So cell wall, what is cell wall? So cell wall is the most important. That cell wall gives the definite shape to the cell and then mostly situated between the capsule and the cytoplasmic membrane that is called as the cell wall. So this cell wall having thickness around 10 to 20 nanometer in thickness and they constitute around 20 to 30% of the dry weight of the cell. And this cell wall cannot be seen by the with the help of the direct light microscopy and they does not stain easily by different staining reagents. So we require the differential staining, plasmolysis, microdissection or electron microscopy or we can see by using the specific antibody. So here you can see the structure of the cell wall or components of the cell wall present in that. So in that we can see so this blank circle that is the N-acetylmuramic acid. Short form is the NAM and this dark circle is called as the N-acetyl glucosamine, that is the NAG, and this uh, vertical line in that small horizontal line is called as the tetrapeptide chain, and this horizontal line in that short um, vertical line is called as the pentapeptide bridge. So these are the main four components of the cell wall. So in that also contains the acid, like first acid is the diaminopymedic acid. So diaminopymelic acid is short form is the DAP, then muramic acid, and third is the teichoic acid. These are the three important acids that contains in the cell wall of the bacteria. So this teichoic acid mainly essential for maintaining the rod shape shape in the rod shape organism, and these substances are joined together and they give rise to the complex structure that is called as the peptidoglycan layer, or the murin, or the mucopeptide. And these peptidoglycans, they consist of the three main parts. So first is the backbone, second is the tetrapeptide side, and third is the pentapeptide side. So in the backbone, they are composed of two acids. First is the NAG, that is N-acetyl glucosamine, and second is the N-acetyl muramic acid. And in the tetrapeptide side chain, they are attached to the N-acetyl muramic acid, and the pentapeptide, their cross bridges are present. So this structure or this backbone structure is same for the all bacterial species. Here you can see the peptidoglycan monomer. This is the structure of the NAM, that is N-acetylmuramic acid, and NAG means N-acetyl glucosamine. And here you can see these are the amino acids that present on the in the pentapeptide bridge. So next, last point we will see in the part 1 video is the function of the cell wall. So what are that? The cell wall is involved in the mainly growth as well as the cell division, and most important, it gives shape to the bacterial cell. So, and third most important, the protection to the internal structure of the bacterial cell, because it acts as a supporting layer, and they provide the attachment to the complement system, and they shows the resistance to the harmful environment of the environment. It means they protect to the bacterial structure. So these are the function of the cell wall. सो दोस्तों इस वीडियो में हमने देखा है कि व्हाट आर द पार्ट्स ऑफ द या फिर स्ट्रक्चर ऑफ द बैक्टीरियल सेल. इसमें हमने चार पार्ट्स कवर किए. फर्स्ट इज द फ्लैजेला, सेकंड इज द पिलाई, इट इज आल्सो कॉल्ड एज फिम्बरी, and third is the capsule, and fourth is the cell wall. In the part 2 video we will see the next part of the bacterial cell. Thank you all.